So let's start with today's session. So here is your first question. Option D. Okay. Anyone else? Yeah. A. Okay. 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 So let's firstly understand the question itself. Which of the following produces dikaryotic phase in basidiomycetes? Firstly, basidiomycetes is a class from the fungi. So it's a fungal thing. Then let's understand the word karyo. When we use the word karyon, karyon is referring to the nucleus. So if you remember when we learn about the cell division cycle, we divide cell division cycle into two things. One is a karyokinesis and then another is a cytokinesis. So in karyokinesis, what happens? The nucleus will be duplicated, means two nuclear conditions will be firstly created where the genetic material will be divided into two segments by doubling the thing, and then in the cytokinesis the cytoplasmic content and other organelles were distributed among it and the two cells will be formed. So here we have to consider about the dikaryotic phase. So that means there is some phase where the two karyons, means two nuclear conditions must be there. So two nuclei should be there. So the conditions that are there. So karyokinesis is definitely not the option, because in the karyokinesis, though the two cells are forming, they are not remain in the same cell. They will be distributed into two cells which will be formed. Then in the karyogamy, what happens is the fusion of the two nuclei. So if the fusion of the two nuclei is happening, that means that is the part where the one nucleus will be ultimately developed in the single cell. So the karyogamy cannot be the thing. Plasmogamy is a specific characteristic which is happening for the basidiomycetes, where the two vegetative cells get fused, means two different kinds of cells get fused, and there are the cell after fusion will be formed have the dikaryotic phase because they have two nuclei. So only plasma membrane contents will be gets fused, the cytoplasmic content will be shared with each other, and two nuclei will be remain as it is. So that is what we call plasmogamy, which is a dikaryotic phase of the basidiomycetes. And apogamy is a kind of a asexual reproduction where gametophytes will help for the formation of the sporophyte. So sporophyte is a asexual way of reproduction and gametophyte is a sexual way of reproduction. But in the apogamy, gametophyte will help for the formation of the uh, sporophytic kind of a plant. So that is not the option. Okay, so for this question, option B will be the correct one. That is the plasmogamy is a dikaryotic phase. So I purposefully for 2019-2020 there is much more things which will be need to be explanation. So few parts I have purposefully included. So you can take a better look after this. I have already explained it, but this is the additional information that you can refer once I share the presentation. Okay. Second question. Basidiomycetes is a class of fungi. Anyone? Anaka, do you remember this part from morphology of flowering plants? We talked about the It's C. Oh, sorry. I didn't get the answer. Sorry. Yes, Gamocephalus is the correct answer. So this question from the morphology of the flowering plants. So among that they have discussed, this is pretty self-explanatory part. So if possible, please go through this topic. because this is another medium weightage topic as per previous question. Therefore, the among the topics that is covered between this chapter, the floral formula, that is the from the last part, is the another important topic that I would suggest to cover, because it's a floral formula, means it's the universal way to explain particular characteristics of the flower. However, to understand that, you know, need to know first flower. So K is the sign used for the sepals, and C is the sign used for petals. So sepals is the green part, which is when we see the bud, there is a green petal-like thing which is covering the bud, and then once the flower is open, we have the petals over it. So K is a representation of the sepals, and then the 5 is the number, and when it's bracketed, when they are joined together for the structure is formed, that is what we call the gamosepalous, that is the sepals are fused to form that particular structure. So bracket is the representation of the fusion, and 5 is the number of the sepals. Similarly, the polysepalous, the sepals are free, like you can see in case of the rose. So they have the separate sepals. And gamosepalous, one example you can see the hibiscus flower, jasmandi that we call, that is the example of the gamosepalous kind of a thing. So option C is the correct one. Next. Sorry, if somebody said something, okay, option B, Roshan said option B, okay. Option D, okay. Option A, okay. A, okay. So these are all kinds of, especially the word, we have this kind of expression in the structural organization in animals, that is the different kinds of tissues were discussed. So blood is a kind of a connective tissue. This is a special connective tissue which is fluidic in nature. That is the one characteristic. And blood is a connective tissue which does not secrete collagen, which is the correct one. Collagen is a part of the bone, and that the structural organ for the muscular and the bone structure, that part will be covered under that. So the option A is the correct one. Blood is a connective tissue which does not secrete collagen. Tendon is a type of a dense kind of a connective tissue, that is correct, but that is a regular kind of a connective tissue. There are two kinds of a dense kind of a connective tissue. One is a irregular one and another is a regular one. So in the regular means the shape, it depends on the shape. So regular shape is present in case of the tendons and ligaments. However, the irregular kind of a connective tissue with a dense nature is the skin. So skin is a classic example of irregular. So that's why the second part is the incorrect one. Adipose tissue is an example of fluid connective tissue. No, adipose tissue is not a fluid kind of a connective. Blood is the only fluid kind of a connective tissue, and adipose tissue have the is a loose kind of a connective tissue. And saliva is secreted by exocrine gland, not the endocrine gland. So hormone secretion, that other part that we have, the one with the endocrine gland, the ductless glands and the duct-free glands, two kinds of a thing. So saliva is a kind of a exocrine gland. It's not the endocrine gland kind of a thing. Next. A, okay. Option B, B, Okay, so this part we have done in the enzymatic system. So these all are the parts for the coenzymes which are non-proteinaceous in nature and but it helps for active functionality of the enzyme system. So hem is the part of the hemoglobin that we have talked, and this is a special guy because these are the tightly linked with the apoenzyme part, and then it helps for functionality. So once the inorganic or non-proteinaceous thing or the organic or the inorganic thing which is tightly linked to the enzymatic, that is the proteinaceous part, such part we call the prosthetic group. So A will be paired with 4, that is the prosthetic group. NAD is a kind of a coenzyme. So these are the coenzyme system, again organic in nature, but they are not much tightly linked. They are kind of loosely linked. They will be play as a transient phase for the intermediate for the during the activation of the enzyme. Zinc is a kind of a cofactor. That is the another class of coenzyme that we have discussed. That is which is the metal ions, which are important for the functionality. They bind at the active site of the enzymatic system and for enhancing its functionality. So zinc is a one of the example work as a cofactor for proteinaceous enzyme called carboxypeptidase for that. Then niacin is a class of the belong from the B complex group. So niacin is a vitamin. Niacin can also work as a coenzyme. Many vitamins can work as a coenzyme for many biological processes. But however, for this particular thing, that will be paired with as a vitamin because that's the only example as a vitamin. For by considering this answer, the option B will be the correct one. Next option, sorry, flagella. Okay, yes, yes, correct. Bacterial motility is done by the help of the flagella, which is a tail-like projection. Next, option B, A. Okay, stroma and intermembrane. Okay. So if you remember the chemosmotic hypothesis, we talk about proton gradient in that particular part. So if we talk about the structural part, there will be the chloroplast membrane firstly. Then there will be on the membranous part, there will be the light reaction will be happening, means the centers for the light reactions will be present at the thylakoid part. Okay, so the thylakoid will be there, and in the so if this is a part of the membranous structure and the thylakoids will be there, H plus ions will be accumulated in the lumen of the H plus ions will be accumulated in the lumen of the thylakoid. Firstly, the thing will be happening over there. So light reaction will be formed in the membranous region where the PS1 and PS2 will be located and the proton gradient will be formed inside. So after that, once the ATP generations will be happening, the dark reaction, that is the which is depend on the products which will be synthesized during this part, will be utilized. Then stroma will be the liquid part. Okay. So stroma, this is a thylakoid. This is the one thylakoid that we are referring to. In the lumen, the proton gradient will be developed and they will be eventually passed to the stroma region to get utilized, and the carbohydrate synthesis will be happening in the stroma region where the enzymatic systems which are required for the dark reactions are present. So that means the proton gradient is generated between stroma and thylakoid lumen because firstly they get saturated inside the thylakoid and then the things will be passed. The proton gradient generation during the respiration, if we are talking about the mitochondrial membrane, then mitochondrial structure, there will be the intermembrane part will be play important role because the large part will be get accumulated and ATP generations will be happening at that time. So please don't get confused. The intermembrane space for the chloroplast won't be the case for the the proton gradient. It is a thylakoid lumen where the PS1, PS2 will carry out because the centers are located in that part. So the thylakoid lumen is the one part which is very much essential to develop that those protons, and then by photo oxidation, and then we will get all the reactions, and the ATP utilization for the carbohydrate synthesis will be happening in the stroma. So it between the stroma and thylakoid lumen the proton gradient will be generated. So that will be option C. Next, C or D. Okay. Option A. Okay. So this is this part from the locomotion where the we see the how the muscular activity is taking place. So it plays important role for the myosin, ripomycin, and all will be play important role. So if possible, I will recommend from that particular chapter, this part you can focus to see how the neurotransmissions will be happening and how muscular transmission will be happening. So from that chapter, please focus on this particular part, the mechanism, how it is happening. So generation of the action potential of the muscle in one, yes. So for this particular thing, the first option will, we are talking about the muscular contraction. So for muscular contraction, action potential is very much key required. So key requirement, so the generation of the action potential in muscles is very much essential to trigger the release of the acetylcholine in the synapses. Otherwise, how the contraction will be taking place? So option A will be the essential one. Second, release of Ca2 plus, that is calcium ions in the from the sarcoplasmic reticulum. Yes, that is another very important action for the activation of the neural muscular junction contraction. Then how the action will be taking place. Ca2 plus, that is the calcium ions, are very much essential for this particular activity. Binding of ATP molecules in myosin head is the thing that we should consider, because option D, shifting of the tropomyosin to expose the myosin head site of the actin filament, is the correct one. Then only the sliding action will be taking place. So that is the correct part. Binding of ATP molecule in the myosin head is the incorrect one, because it's not the binding, it's the dissociation, because this is energy required process. Contraction is happening at the cost of ATP. So here the dissociation of the ATP molecules to myosin head to the actin site, it what happens, ATP will be dissociated into ADP and phosphatidyl, which will be eventually generating the energy for utilization of the muscular action. So it's not the binding of ATP, it's the dissociation of the ATP which is happening at the myosin head on the once they are sliding on the actin filament. So option C is the correct one. Next, option B. Option A. Okay. So this is a typical electrocardiogram question, which is another part of the circulatory system. So please go through circulatory system, how blood circulation happens, one part, because that will include the structure of heart and how where the all the things are needed, and basic structure of the ECG and how things will be happening. So P will be the part, the two nodes are there. One is a SA node and another is a AV node. AV is a atrial ventricle node, that is the one part. Then the sinoatrial node is the another part. So first the sinoatrial node will be contracting, which is the important part for the pumping the things in atria, which where the P part will be generated. Then ventricles get contracted, that is a part of the systole. Systole and diastole, two things. Systole is the contraction part of the ventricle and diastole is the relaxation part. So when the AV node get contracted, the functionality of the AV nodes, the work out, then the ventricles get contracted, which will be helpful for generation of the systole. And once the which is the represented by QRS complex. Here the initiation of the systole, it's the peak and this is the end of the systole. After that, once the ventricles get relaxed again and which is represented by T. So if we are saying the AV node is not functional for some or other way, there will be no contraction will be taking place because the AV node is the muscular part which is attached to the ventricles, and if the muscular part is not functioning, it's not functional at all, then the QRS complex, that is systolic contraction, will not take place. So that means in the electrocardiogram you cannot get QRS complex. So option B is the correct one. So QRS complex will be absent if the AV node is not functional. Next. B, 1 and 2. Okay. O- Option B. Okay. So we want clonal progeny from with the same genotype from the mother plant. So that is means the only single parent will be involved for the formation of the clonal progeny. This is the first one we should understand. Then the definitions that we can see over here. Parthenocarpy is a kind of a formation of the fruit without fertilization. That is a clear understanding. So that means only here fertilization is not happening. That means only one parent will be there. Two gametes are not present. However, in case of parthenocarpy, such fruits do not have any seeds and thus no progeny will be formed. So if no progeny will be formed, then how it will be spread to the clonal proliferation? So though only one plant or is involved for the parthenocarpic fruit to develop, as the progenies are not formed from this, we can question for this particular question, this cannot be the answer. Then syngamy or amphimixis. This is a sexual way of reproduction where progeny is formed by fusion of the male and female gametes. If fusion will be happening, then progeny cannot have the same genotype as mother and mother plant only, right? There will be fusion of the parent plant also. So syngamy is definitely not the option. Apomixis is a special mechanism where the seeds are formed without fertilization, and this is a apomixis is a special kind of a form where the asexual reproduction mimics the sexual reproduction. Means it feels like sexual reproduction, but however, the seed formation happening by asexual way. So this means the progeny formed with this particular mechanism has genotype similar like a mother cell because no fertilization will be happening, and seed can give rise to the next plant. Means they are not sterile in nature, we can say. And progenies are not, because parthenocarpy no seed will be formation. In apomixis seed formation will be happening, but this is without fertilization. So apomixis is the one part, and the polyembryony. Polyembryony is where as the name itself suggests, poly is the many and embryony is the many embryos are formed. So embryos will be ultimately lead to formation of the seeds. But in the polyembryonic culture, what happened? This is the embryo can be developed from the nuclear and integument cells. These are the additional parts present inside the cell. So here the genetic nature of the embryo is deployed in nature, but which is identical with the mother plant. Though embryo formation will be happening, this embryo development is happening from the nucleus or integument cell of the same plant. So that's why polyembry and apomixis cells, the embryos that or the seeds will be formed by this method will be have the genetic makeup same as the mother cell. So if this is a if this is a case, we are talking about polymixis and polyembryony. So 3 and 4. So option A will be the correct one. Next. C. Okay. C. Okay. Why do you think it's C? Mm-hmm. Okay. How about mitochondrial? If you look at this, this is a very classic case that you can see before. Firstly, for such questions, you can firstly look for the rule out things. Okay. So you are seeing the mother is infected and the both in the next coming generation, both parents means father is either heterozygous or homozygous. You don't know the things over here. But violent is definitely not case as the females are getting infected in the process. So definitely you can rule out that. Other different option that you can see is the mitochondrial one. Mitochondria DNA is only transmitted from the female to its progeny irrespective of sex. So either it will be male or the female. So if this is the case, the classic case, that first thing that you can remember is the if the mother is have the mitochondrial disease or certain genetic associated with the mitochondrial DNA, that will be getting by the all the children develop from it. So you can see in the first generation, all children have the disease. Okay. Secondly, mitochondrial DNA, the disease will be transmitted only from female. So if the male is infected, the disease is not transmitted to the next generation. That you can see over here. Male is infected, the parent from the male is infected, however, the children are normal because the female is normal. However, when the infected female is there and it get married, the next progeny, irrespective of sex, have that kind of a disease. So this is a very, very classic example of mitochondrial, means before ruling out X recessive dominant more than this thing, you can see this is as a classical example of mitochondrial transmission of the disease. If the female is infected, first generation you will get all children infected definitely, and then male affected children cannot able to transmit the disease unless they are marrying with the female who has the disease, like so. Or on the other hand, if the female has the disease, then it is passed to the female child, and irrespective of the sex, the children are infected. So look at the pattern, means before ruling out, such pattern is a very unique example. So this is a case of the mitochondrial disease transmission. Next. Option D. So here we are talking about total 80 minutes of generation time, means total 80 minutes of cycle. That means 4 generations will be happening for E. coli cell. Firstly, they are all labeled with N15. Okay. Then the first cycle will be taking place after 20 minutes. We will be having, because the growth medium is N14 contain in medium. So that means the next progeny will have the half. So half will be have N15 and half will be have N14 because two strands will be there. So that means they will be completely 100% we will get after the first generation, we will get the 100% hybrid progeny. In the next cycle, means after 40 minutes, we will have both the strands. Means we have N15 number of strand and 14 number of strand. But however, the new strand that will be formed, that will be have N14 only. So that means at that time, in the second generation, we have 50-50 chances for light and hybrid kind of a chain. Then in the third generation, Again, N15 strand will be there, but its percentile keep on reducing because the N14 number of strands will be on the higher side. So that means then we will have in the third generation 25 hybrid and 75 light. And after 80, we will have only two strands with the which have the means two sets with only the hybrid nature. However, all new sets that will be formed will be from the means the amongst the 16 strand, two strands will have only the hybrid nature which will have a N15 and N14, and all others have N14, N14. So that means 87.5% light and 12.5% hybrid. So option D is the correct one. Uh, I think it's pretty clear because if you are not seeing the axis representation, I think that's everybody has problem to see the diagram. Parameters in the question we have the parameters. X-axis represented body size and y-axis represented frequency. Option C. Okay. That dash light represent the ancestral one, and the continuous line will be representing the distribution after few generations, mean the new generation you can say. C. Option C. Yes, option C is the correct one. So this question belong from the class topic of the chapter evolution. And amongst this, the Hardy-Weinberg equilibrium is the another important way for the distribution. Please do this topic very thoroughly because this is a part of interest for the all the scientists who are working in the evolution in the IISER. This is their favorite topic. So they will ask question based on this. We will find this particular topic discussed in many other papers also, either the equation that have based or the way it is distributed. So this is from the part of the evolution. So please do focus on that topic. So here we are talking about the size distribution of the fruit fly generation. So dashed lines will be representing the ancestral distribution and the continuous lines will be representing generation after that. So body size, as the body size keep on increasing, that is the way of the, so here they have the better survival rate and the better reproduction rate. So because of this, they are more naturally get selected over the other generation. That is the case. So if this is the case that we are talking about, the ancestral generation will be having the means from lower, medium to higher, that kind of a thing. However, they get more favored, so that's why the distribution is like this. The higher the body size, they get more selected over the earlier generations. So because they have the better survival rate. So that is very much well presented by option C, which is the correct one. Next. Option D. Okay. Okay. So option D is the correct one. Two things. One is the active immunity, another is the passive immunity. So active immunity, what it happens, we are getting exposed to the antigen or purposefully introduced. Naturally, we will get to the in contact with different kinds of antigen. Either it will be the disease or different kind of a thing. and then our body helps to develop the antibodies as an immune response against it. That is the way of the active immunity. And secondly is a passive immunity, where we introduced the directly antibodies, different kinds of antibodies, and as a kind of a treatment or as a kind of a natural process that can happen. So this is a, without exposing to the antigen, we are directly introduced by the antibodies. That is a kind of a passive immunity. So transfer of antibodies from the fetus via placenta. Here naturally mother is transferring some antibodies to the fetus. So this is a natural way of the passive immunity. Here no exposure to the antigen. Directly antibodies are getting transferred by the placenta. So that is a passive kind of a immunity. More specifically, naturally happening passive kind of immunity. In vaccination to infants, we purposefully introduce weakened or dead bacterium, viruses, any kind of a thing to the infant, so our body can prepare antibodies in advance. So in future, if the exposure will be there, the body will be taking care of such infection by the developed antibodies based on its memory. So this is artificially introduced active immunity. So option D is the correct one. Next. Option A or D. Okay. Option A. So PCR is temperature dependent cycle where the replication or the point of our interest will be get replicated, right? So firstly, the two double-stranded DNA should be get separated, which will be happen by the process of the denaturation. So that means which is happening at 98 degrees Celsius. The temperature will be at 98 for the 30 seconds or 10 seconds even. So the two strands will get separated. Then the primer should be get annealed. So the primer temperature will be very important, that melting temperature. So that will be the lower temperature, which will be ideally between 50 to 55 or 58 degrees Celsius. So the annealing will be happening with the primers at the given locations, and then the extension will be happening, which will be again little bit higher, that is 72 degrees Celsius, but not as high as the denaturation. So extension will be helping for polymerase to work to add the complementary bases and make many copies of DNA of interest. So denaturation, annealing, and extension are the steps for the polymerase chain reaction. So option A is the correct one. Next. Option A. Yes, option A is the correct one. These are the four different ways by which the biodiversity get threatened because of the many reasons. And carrot grass is a example of the alien species because of its invasion. In the localized species cannot grow very easily. The nutrition depletion will be happening for those species. So this is the alien species invasion case. So option A will be the correct one. So it's already actually 11:50. Do you all have time to solve the next paper? Is it okay if you? Okay.